Our guests represent well-known universities and institutions in Europe.
These trees are probably 5 years old and they're all dying. We know not what is causing it.
At this training school I'm presenting a general overview of what tree diseases are coming into Europe as invasives from other continents, and I'm also presenting some information on a potential alternative pathway for spread of these alien invasives.
We're not very good at what we call preemptive prevention. We're not actually that good at stopping future pests from coming in.
We're here, in fact, to have a training school for that cost action, dealing with identification of pests and pathogens that might be moving in the trade. Our main aim, of course, is to understand why those pests are moving and how we can actually bring new measures in to prevent the movement in the future.
I expect to give the students quite a wide knowledge of certainly the pathogens that are affecting trees in Europe and the ones that are not here yet but are threatening to come in. There's also the problem of ones that have established in small regions of Europe and are likely to spread out from there, cause problems probably everywhere.
I would like to apply the next generation sequencing strategies for the detection and identification of the pathogens, to help them in the beginning to get used with these technologies, to learn to them accurately how to use these strategies, and mainly to discuss with them the limits of this strategy. It's very important to show the limits and where this strategy could not be applied.
The students will come back to their institutes and will be able to use online databases and how to analyze DNA barcode data for an efficient and an accurate identification of invasive and pest species.
This tree shows the typical symptoms of Phytophthora disease with a rarefaction of the crown. And if you look to the stem, it's a clear depression. If we can debark the plant, we will see a flame, a characteristic flame. That is, I think, this is the main symptoms that can help us to distinguish plant that is damaged by Phytophthora. On the back, you see other plants with symptoms that are less heavy but are clear, belong again to Phytophthora attack. And compare with the healthy plants, it's really easy to recognize symptoms. The problem sometimes is when you have only diseased plants, because you know that other pathogens can cause the same symptoms. In this case, the necrosis are not deep as for chestnut, for example. So this could be a necrosis caused by Phytophthora. So the idea is to bring back this in the lab, clean this, wash in water, dry, and then put in the selective medium, taking this part that is between the brown and the white part. The early detection is one of the main steps to prevent the introduction of alien invasive species in Europe. I'm here to share my experience on the detection tools that can be used to identify Phytophthora, with attention to traditional methods, molecular methods, and especially the new generation system approaches. Another way to identify Phytophthora, we can use a serological test. These tests are very useful because can be easy used in the field. It's like a pregnancy test. There are two lines. One is for control and the other one is for the sample.
The most important thing for me was to learn how how to describe certain types of Phytophthora, how to inoculate plants, how to use different methods, because we were using different methodology, also the antibiotics they're using.
I am early stage researcher and I have to learn a lot of new techniques, isolation and molecular techniques. So it's really useful for me, this cost action.
In this training school I teach on the use of real-time PCR to assess, to prevent the pathogens in forest.
The most interesting for me is a PCR reaction, and I learned a lot of new things, and I could talk with the expert, and it is very nice and it is very helpful for my research.
I'm here to try to raise the attention of the trainees on the pathways of arrival and spread of invasive forest alien pathogens.
My wider expectations is to get people interested in working in this field, because we really need people to work in this field.
This is very important to know how the process in Europe is organized, and to create also kind of networking across Europe.
We have 29 trainees from 16 different countries around Europe, and they're mainly what we call early stage researchers. These are scientists who are new into their science careers, and they're here to learn about the movement of pests internationally.
It is really nice. You can meet new people from all over the world.
Cost is a really nice opportunity for us, and I'm a real example of achieving cost goals in cooperation in science and technology. I'm very happy to be here.
The school is fantastic, yes, very friendly scientists, so in such environment you can enjoy it, so you're not worried that you'll say something wrong, because everybody's feeling very happy and they are really eager to help.